Hello everyone, my name is Nadine Kurz. I'm from the Department of Medical Bioinformatics at the University Medical Center Göttingen, and today I'm going to present AdaGenes, a variant annotation and filtering pipeline for whole genome and whole exome sequencing files. AdaGenes comes with unique new annotation features and is both usable from the command line and our interactive web interface. So let's take a look at the different types of genetic variation and how to interpret them. Here we can see a rough overview about the main variant types we can find in the human genome. The most frequent variants are single nucleotide variants, where a single base is substituted by another in the DNA, which can result in an amino acid substitution or a stop codon. We then have small insertions and deletions, where small parts of the DNA are being cut out or added, which may result in a frameshift, destroying the rest of the protein. In case of copy number alterations, we have parts of the DNA being deleted, copied or duplicated at another position. And finally, we have gene fusions, where two genes are fused together. In all cases, the variation in protein coding variants may result in an altered protein or no protein at all. To actually evaluate the biological consequences of these variants, we collect different kinds of information, including the population allele frequency, pathogenicity predictions, the functional regions and the gene expression of variants. We also calculated the change in molecular characteristics, the structural alteration, the change in the binding site, and the change in protein stability. Here you can see how this information is actually used to improve the treatment of a cancer patient. In a molecular tumor board, we have a tissue sample of a patient, which is sequenced, and the variants compared to the reference genomes are identified. The variants are then annotated and interpreted, and the results are then discussed in the MTB conference for each patient individually. The MTB finally makes a treatment recommendation for patients based on their molecular profiles. To support the variant interpretation process, we have developed several tools to support variant interpretation for clinical usage and for biomedical research. The base for our variant annotation is a large set of annotation modules, which are all independent web services and can be accessed with an application programming interface. To efficiently annotate and filter whole genome files, we developed AdaGenes. After a small set of pathogenic, clinically actionable variants has been selected, the clinical variant interpretation toolkits MTB Report and Oncopus can be used to find targeted therapies for cancer patients. And here you can see the architecture of AdaGenes. The tool can be used as a standalone Python package from the command line or as a web application combined with a NoSQL database. The web frontend is implemented using Vue.js, while the server is a Flask service using Python. AdaGenes supports several data input formats, including VCF, MAF, or custom CSV files. In case your data is in a different format than VCF or MAF, AdaGenes can actually transform variant data into VCF. In addition, it supports liftover between reference genomes. The annotation is accelerated by splitting large files into chunks, which are parallelly annotated and merged afterwards. In addition, the chunks are further split into HTTP requests with 100 variants each, which are then sent to the annotation modules. Finally, the annotated data can be sorted, filtered, and written out again in multiple file formats. To annotate multiple variant types, we implemented a type recognition before each annotation process. AdaGenes then annotates the variants according to their types, including basic variant information like the transcripts, protein, and cDNA identifiers, pathogenicity predictions, and population allele frequency data for SNVs, molecular and protein features of the mutated sites, drug-gene interactions, and gene expression. For copy number alteration and fusions, we also analyzed breakpoints of the DNA. Since one of our goals is to understand the biological fundamentals of pathogenic variants, we implemented a couple of novel annotation features for protein coding variants. We dynamically compute the characteristics of mutated sites within the main select transcripts of a protein. We used protein structure predictions computed by AlphaFold and computed several features, including the relative accessible surface area, the alpha carbon atom distances of each position relative to the mutated site, the protein domains, and the secondary protein structure. To analyze the consequences of variants at a molecular level, we computed a change in charge, hydropathy, aromaticity, polarity, and the ionization and phosphorylation potential. And this is how the AdaGenes web interface looks like. We have an upload into input format on the left side, where we have two submenus, annotations and transformations, which will be activated depending on the data you upload. On the main menu, you see the variant table, which can be sorted and filtered and interactively browsed. And this is how annotation process works in the web frontend. After selecting the file you want to upload, you can define the reference genome of your variant file, as well as your desired output reference genome. In case you select a different genome, AdaGenes will run a liftover and convert the genomic positions to the new reference genome. When you click on Upload, AdaGenes will pause your uploaded file and display the data as an interactive table, where we have two kinds of columns. On the one hand, we have the standard VCF columns, including chromosome position, reference and alternative allele, as well as each annotation feature in the info column as a separate column. You can then select one or multiple annotation features, or simply select all of them. If you then click on Update Data, AdaGenes will annotate the variant data and generate a new column in the produced table for each annotation feature. Once the annotations are displayed, you can interactively sort and filter your data according to the new features. For numeric features, you can create complex queries combined with AND OR operators. For text features, we have several options to create string-based queries. In addition, you can simply sort the variants by clicking on the column headers, which is a lot easier than defining your own filtering options from the command line. Another novel feature of AdaGenes is its ability to transform variant data from transcript and protein level to genomic level. For example, in clinical practice, you often have datasets that describe variants at the protein like KRAS G12C or BRAF V600E. If we want to annotate this data, we have to transform it into a VCF, which means mapping it to the genomic level. For this purpose, we have developed CCAT, or Sequence Conversion and Analysis Toolkit. With CCAT, we can map variant data from transcript or protein to the genomic level and vice versa. We're actually doing this by mapping variants from the protein to the genomic level based on the main select transcript of a gene. With AdaGenes, you can do this quite easily from the web interface using the same upload form as for the VCF and MAF data. You can simply upload a file in CSV, TSV or Excel data format. AdaGenes then parses the data and simply displays the uploaded table formatted data. Since the tool recognizes that it's not a VCF or MAF, AdaGenes then activates the transformations, and you have now the option to convert your data into VCF. In this case, you actually select the columns of your data which represent gene and amino acid exchange, and AdaGenes is then able to produce a VCF at the genomic level, which can then be downloaded or annotated like a normal VCF file. In addition, we provide several analysis and plots, including the mutation types and distribution, as well as the predicted pathogenicity scores of the most pathogenic variants. These are machine learning-based methods and provide a pathogenicity prediction, which is usually between 0 and 1, where 1 represents a pathogenic prediction. In addition, you can analyze the genes with the highest number of mutations. Here, the size of the circles is calculated as the average alpha-missense predicted pathogenicity of all variants within a gene. And in this example, we see a high number of mutations in ANAC2, but which are mostly predicted as being benign, while we have a high number of possibly pathogenic variants on TP53. In addition, you can also check out the protein domains where protein encoding variants have been identified. After you've annotated and filtered your variants, you can export your data in multiple file formats, including VCF, MAF, or CSV. In addition, we provide a log file which records all the annotation and filtering steps to ensure reproducibility. After you have annotated and filtered your data with AdaGenes, you can go on with analyzing the remaining, possibly a small set of pathogenic variants in detail, using our interpretation frameworks MTB report and OncoPus. Like AdaGenes, OncoPus provides a state-of-the-art web application which is specifically designed to analyze the clinical significance of variants in cancer. In OncoPus, you can either upload a variant file or directly search for variants. In addition to the variant table, you then get a list of possible personalized treatments, the proven evidence of their clinical significance, as well as interactive visualizations. In OncoPus, you can analyze each variant in detail by clicking on the detail page of a variant. What you see here is just a rough overview about the different tabs of a variant page. For example, for SNVs, we provide genomic and protein variant details, clinical data about possible targeted therapies for a patient, pathogenicity predictions, as well as an analysis of the structural context of the protein of a variant. So let's take a look at how we can actually use the protein annotations of AdaGenes to interpret unknown variants. Here we have 4 examples of variants, which are all labeled as variants of unknown significance in a ClinVar database. OncoPus provides a visualization of the whole protein, including known pathogenic nearby variants, the relative accessible surface area, the alpha-carbon atom distances from each amino acid in the protein relative to the mutated site, protein domains, as well as the secondary protein structure. In addition, we have a 3D visualization of the main select transcript, which is colored according to the average pathogenicity prediction by alpha-missense at each position of the protein. And in this example, we have a TP53 variant, which is located in a loop structure and outside of the protein domains, with a rather high surface accessibility and a benign prediction by alpha-missense. In contrast, we have a BRAF and an EGFR variant, which are located in alpha helixes and protein domains, with a pathogenic prediction by alpha-missense. In addition, we can use the alpha-carbon distances to find potential targeted therapies for nearby variants. In case of the EGFR variant, we actually have a variant at position 785, which seems to be closer to the mutated site based on the protein sequence. But if we take a look at the folded structure, we can actually see that the variant at position 719 is closer in the spatial protein structure. In addition to the protein analysis, we created an innovative sunburst plot to browse possible targeted therapies for cancer patients in OncoPus. For each plot, we are visualizing one or multiple variants of a patient in the center, while the outer layers represent characteristics of the clinical evidence of possible therapies. In this example, we have a drug classification in the first layer, the possible drugs in the second, the evidence level of the underlying study in the third, and the publication ID of the study in the outermost layer. For example, if a clinician has a specific preference regarding a drug classification, or wants to find out a therapy which has been proven to be efficient for the patient's cancer type, the sunburst plots provide a visual way to browse possible therapies effectively. Since the plot is interactive, you can click on each cell in the plot, which causes the plot to zoom in that cell. Finally, you can click on the publication ID on the outermost layer to directly check out the clinical study for a treatment. Another option you have to create your own reproducible variant annotation and filtering workflows is to use our workflows we created for the framework NIME. In NIME, you have the option to visually create your own pipelines by connecting nodes using drag and drop. The NIME workflows are specifically tailored for data cohort analysis if you want to analyze a group of patients and their molecular profiles. So to conclude, we have created a comprehensive variant annotation and filtering system with AdaGenes, which is easy to use within the interactive web application. AdaGenes is also available as a public instance where data is uploaded anonymized. And with that, I would like to thank our supporters, including the Comprehensive Cancer Center Niedersachsen, the Volksbank-Stiftung, the International Max Planck Research School for Genome Science, and the University of Göttingen, and of course all my colleagues at the Department of Medical Bioinformatics. And with that, thanks a lot for your attention, and you're welcome to test AdaGenes by yourself.